Okay, so thanks for having me, everyone. It's an absolute pleasure to be able to share my current experiences with you and kind of walk you through how I got to where I am today as a PhD researcher. So, in terms of my career progression so far, PhD research is considered early career, so right at the start of my scientific research career. And it's all about conducting independent research in an area of your choice that interests and intrigues you. So before I start discussing the topic for today, I'll give you a bit of a brief background about where I am currently as a PhD student, and then I'll walk you through some of the experiences that I actually went through to get where I am today as just a background. So I'm a PhD candidate and coral reef scientist at the WA Oceans Institute and School of Earth Sciences, as well as the School of Life and Molecular Sciences at the Curtin University of Technology here in Perth. So it's taken me about 7 to 8 years to get to this stage in my career. And what I'm currently working on is what we consider as coral reef islands in the subtropical Pilbara of Western Australia. So I focus on more of a landscape level coral reef research question where I'm trying to reconstruct how islands and their reefs essentially will change with climate change. So islands as a landform are actually built by the surrounding reef system. So if you think of an island like Rottnest Island, for example, that's a continental island. So the islands that I work on are built by reef organisms, so corals, sponges, mollusks, echinoderms like sea stars and urchins. And over time, those organisms actually produce sands, or sediments we call them more technically, that actually accumulate on these reef platforms in tropical environments and actually build an island landform. So if you think of islands in the Maldives, in the Marshall Islands, Kiribati in the South Pacific, they're all built over thousands of years by reef organisms. And so the purpose of my research is to try and underpin how exactly reef productivity drives island growth over time. And if these islands are subjected to the impacts of climate change in the future, we're likely to see a reduction in the amount of sediments produced on those reef environments. So if you think of coral bleaching, decimating reefs, it will have an impact on how much coral turns over in the system. So if you think of a live coral reef, it needs to die and break down through a natural life and death cycle to produce the sediments that build landforms. So why is it important that we preserve landforms like islands? Is predominantly because of their conservation value. They're important as rookeries for sea turtle populations, shorebirds and seabirds, and therefore play an important environmental asset to the conservation value of a region. And in the Pilbara in particular, many of the turtle rookeries for Ningaloo Reef, for example, are reliant on those islands in the northern part of the Pilbara. And unfortunately, the video is not playing, and it's actually a video of these islands. But I can send through some images if you guys have any particular interest in this study area. So I'm just going to give you a brief background as to how I got to where I am. So choosing the right degree is important. And when I was in your place back in 2013, I was thinking about where I wanted to go in my science career. I knew I wanted to pursue a career in environmental science, but I didn't quite know whether I wanted to do a terrestrial or a marine-based research stream. So I was lucky enough to get an early admittance into the Bachelor of Science, Wildlife and Conservation Biology at La Trobe University in Melbourne. So I'm originally from Victoria. And this was a very important degree in my career because it gave me an introductory kind of look at a whole suite of environmental sciences. So I was lucky enough to study everything from botany and plant ecology. I was quite a plant nerd actually when I was in undergrad. So I was really into studying plants and how they interact with environmental factors. And we were lucky enough to visit areas of Victoria, such as the Great Alpine National Park, which you can see down below here, running quadrat surveys, everywhere to the arid zone regions of the Wimmera and Mallee of northern Victoria. I was also able to do subjects that streamed ecological and evolutionary genetics, looking at how environmental factors actually drive the genetic change of organisms. And that ranged from both terrestrial, freshwater, and marine organisms. It wasn't until my third year that I was actually able to take my first marine science stream of subjects. That was in a third year zoology unit. And that encompassed marine physiology and coastal marine and coral reef ecology. And it was doing those units that actually was a turning point where I knew I wanted to pursue a research stream in the marine sciences. So essentially, I graduated with a multidisciplinary degree across both terrestrial and marine systems. So I think I co-majored across ecology, botany, and zoology. So quite a large variety of choice. So my first taste of marine science came in my third year when we were lucky enough to go on a third year field course to the Spectacular, and this is actually one of my favorite regions in Australia, the Heron Island Island system and reef, which is part of the Capricorn Bunker on the Great Barrier Reef. And if you think of Heron Island, we all think of this beautiful tropical idyllic reef system with an island sitting on top of the platform. That's pretty much what you see when you go out to this spectacular part of the world. It's got a high amount of terrestrial and marine biodiversity. And we were able to conduct experiments as part of our third year unit in marine physiology and coral reef ecology. And that was kind of a light bulb moment for me that I was like, okay, I want to actually go further after my degree and do an honours degree and start studying coral reefs. And I'm going to actually share with you a project from that honours degree that I was lucky enough to do also on Heron Island from 2014 onwards. But before we go into the nitty-gritty of the project, we need to know a bit of the basics on exactly what this system is structurally. So what is a coral reef? And we all have been encountering, coral reefs in the media for the last several decades. So we are quite familiar with what they are generally. But scientifically, we know them as large three-dimensional structures that are built over time, often over thousands of years, by what's known as scleractinian corals, or the hard reef-building corals. Now, reef-building corals essentially build the physical framework of the reef, okay? So if you think of that natural life and death cycle I talked about earlier, the death of corals helps to produce the next set of deposition on the reef that builds the framework. And essentially, the reef is growing towards sea level. So as sea level increases throughout time, so does the reef. It builds up incrementally. So what are these organisms that are building the reef? And there's quite a misconception about what corals exactly are. So corals are not rocks. They're actually an animal. So the animal is called a polyp, called the coral polyp. It has tentacles. So morphologically or characteristically, they look quite like jellyfish and sea anemones, and they actually belong to the same family as these two organisms, the family Cnidaria, they're called. Cnidaria meaning organisms that possess stinging cells. So just like jellyfish sting, so do corals. They actually compete with each other by firing their stinging cells to compete for space on the reef. So the coral is an animal, but it also forms a very important relationship with another organism, and that actually happens to be a plant. It's an alga, so a single-celled algae from the genus Symbiodinium, and we call this algae zooxanthellae, or a dinoflagellate. And this actually is housed in the tissue of the coral. So if you think of your skin cells, for example, imagine housing a plant cell within your own animal cells. And that's exactly what the coral animal is performing here. It provides a habitat or a shelter for the algae and protects it. And then in return, the algae provides the animal with a source of food. And that source of food is derived through photosynthesis, just like land plants. So the algae is producing glucose, a sugar, which then supplies the animal, polyp, with a source of nutrition. And the coral animal can therefore perform basic functions like cell differentiation, which helps the animal to grow, just like we grow and extend and replicate our cells, but it also helps the coral animal perform other functions like reproduction. So it actually produces this beautiful, neat symbiotic relationship. Now, in saying that, where does the framework come from? Well, the combination of algae and animal and its biology is actually precipitating the skeleton that we see building the reef. So over time, as the coral animal is growing, it is also depositing calcium carbonate skeleton around it as like a shelter, essentially. And that helps to protect the animal and the algae from external predation and environmental parameters like UV radiation. So as the framework starts to grow towards sea level, it's depositing this skeleton around it as a form of protection. And that's what's building the physical reef that we experience today. So this complex three-dimensional framework actually provides habitat for 25% of all known marine species that are found on coral reefs. Everywhere from your other vertebrate species that we see, from the herbivorous fish, low-order carnivorous fish, up to our top-order predators. But it also provides a complex habitat for invertebrates. So echinoderms like urchins and sea stars, they rely on the complex framework of the reef for shelter. And of course, general foraging on other forms of primary producers like algae, which also grows around the coral colonies. So they're very complex and rich systems, otherwise known as the rainforests of the ocean. So more in terms of a human benefit, reefs worldwide provide up to $30 billion in economic benefit. So if you think of where coral reefs are located globally, they're usually confined to the subtropical, equatorial, tropical regions of the planet. So a lot of the coastal regions and coastal nations rely on coral reefs heavily in terms of food and fisheries, providing a source of seafood and income. And more locally, they actually produce a form of tourism dollar. So just like here in Australia, our Great Barrier Reef Marine Park and the Ningaloo Reef Marine Park bring in a lot of tourism dollar, so supporting the economy from a much more different side. But we're all too aware that in recent decades, we're in a climate crisis. So coral reefs are under increasing threat from a number of different environmental factors. And we're going to explore these in the presentation today. So one of the most pressing ones is changes in ocean chemistry, and that changes the amount of those chemical ions which are in balance in the ocean. So if you think of a lot of the coral reef organisms that build shells and skeletons, changes in the acidification of the ocean can sometimes throw out the balance of how many carbonate and calcium ions are present in the water. That can therefore affect how corals precipitate their skeletons, for example, as well as mollusks and how they build their skeletons. So you can have an absolute ripple effect from these drastic increases in acidification. One of the most pressing issues is actually increasing water temperature. Now, with global warming and increasing water temperature, These can have detrimental effects, including coral bleaching and increasing the severity of storm frequency on reefs. We're going to focus on increasing water temperature as the primary cause of coral. So we're going to be focusing on increasing water temperature as the case study for this presentation. To have water pollution, and this is a very dire impact, which is actually affecting parts of the Great Barrier Reef currently. So if you think of agricultural runoff from pesticides, this is likely to end up in freshwater river systems and then be expelled into coastal marine ecosystems like coral reefs. And this can cause an overexplosion of nutrients in these tropical environments, which can lead to such effects like outbreaks in crown of thorn starfish. Lastly, unsustainable fishing practices can have a dire impact in taking out species of fish which are highly reliant in improving the productivity of reef systems. Okay, so now we're going to focus on a case study where I've actually applied research to understand how corals in particular are impacted by a certain climate change scenario. So during that 1-year honours degree that I was lucky enough to do on the Great Barrier Reef, I performed a 6-week field campaign at Heron Island between the years of 2014 and 2015. And if you don't know where Heron Island is located, it's located on the southern extent of the Great Barrier Reef, just off the coast of Gladstone. And this is a cooler part of the Great Barrier Reef, and it forms a group of about 4 islands, including One Tree Island, Lady Musgrave, and Lady Elliot. And these form what's known as the Capricornia Cays National Park. So I was looking at understanding how will the coral organism itself recover under high temperatures as well as injury. Now, when I talk about these two factors, we have to understand they're both intricately linked, and they're both linked to increasing climate change scenarios that we're currently undergoing and we project to undergo in the next decade to 2 decades from now. So the focus of the study was on understanding how corals, particularly in these shallow reef environments here on the reef flats, respond to these two factors. Now, the reef flat environment is a very important environment on these platform reefs like Heron Island, because they actually house very thermally tolerant coral species. So this region of the reef actually is susceptible to a large tidal regime. So during high tide, the maximum depth may be up to 1 to 1.2 meters. But during low tide, the corals could be fully exposed to the elements at a tidal range of about 0.3 of a meter, so 30 centimeters. So choosing corals off this area of the reef, it would be very important in the study that I'm going to talk about today. Let's talk about these two important factors in a bit more detail so that we can better understand how we perform this experiment. So, high water temperatures caused by climate change induce a major effect in corals that we know as coral bleaching. And I'm sure most of you are thoroughly aware of what coral bleaching is, even on a quite basic level. But we'll talk about it and kind of unravel this phenomena in a bit more detail. Now, the image that is on the screen currently, we can see 2 drastically different corals in this image. And we can just discern by the color that something here is oddly wrong. So the coral on the left-hand side, it's a plating coral. It's called an acropora. It's actually got a healthy coral pigment. You can see it's actually naturally brown or browny green in color. And what gives the coral its pigment is actually the alga that's actually in the host tissue of the coral animal. And so the coral is housing the algae in the tissue, and that is actually producing the pigment which gives the corals its natural color. So if you're snorkeling on a reef and you see corals of all different colors, it's usually the responsibility of the algae. Now, what happens is you might think, okay, so how long do water temperatures have to be at a certain temperature to induce stress in the coral? Well, branching coral species like this one, or both of these, actually start to stress at about 32 degrees, and bleaching starts to happen at about 34 degrees. Now, natural temperatures on the reef currently are around 24 to 26 degrees. So between 26 degrees and 34, that's a big change in temperature. So what happens is that in the coral itself, once it detects that temperatures are at that very high threshold of about 34, it starts to undergo a stress response. Now, the algae in the tissue actually starts to produce a toxin that's actually harmful to the coral. And we call those toxins reactive oxygen species. And it's like a toxic chemical like hydrogen peroxide. It actually starts to produce this chemical in very high amounts that the coral animal starts to detect it in such high volumes in its tissue. As a response, it goes, as it would naturally, it starts to reject this toxin and say, this algae is trying to harm my cellular functions. It's preventing me from performing tissue growth that's important for growing the tissue and skeleton, as well as performing calcification of the skeleton. And therefore, as a response, the animal then rejects the algae and it expels it from its tissue. It literally pulses it out into the water column. And that's what we see as coral bleaching. So the white color that we see is actually the tissue becoming transparent. It's lost the pigment that the alga has given to the coral. Now, the coral itself, even though it's rejected the algae into the water, if the temperatures drop within a week of it being at 34 degrees, for example, it can then reuptake the algae from the water because it's then lowered its stress level. It's saying, okay, I'm now happy enough to uptake the algae and start to perform normal cellular functions like growth and reproduction. If the temperature remains too high for too long, above 2 weeks or 2 to 3 weeks, essentially the animal will starve to death. It won't be able to uptake the algae anymore and therefore can't get that glucose or sugar needed to perform growth and reproduction. So it can have quite a dire impact on reefs. The next pressing factor is that climate change induced temperatures can also have a major impact on increasing storm events and cyclones in tropical regions of the planet. So you might think, okay, it's still detrimental that a cyclone comes through and might have an impact on a coral reef presently, but with climate change, we predict the frequency of these storm events to get more regular. And they might not happen just 2 times a year, but they might start happening 3 to 4 times a year. And that can have a long-lasting ripple effect on how reefs recover. So why is this the case? Well, if you think of a normal productive coral reef, most of the species that occupy these reefs, like these plating corals and branching corals, are actually quite sensitive. They have more surface area that's able to be in contact with waves breaking on the reef. Now, storms and cyclones produce a lot of swell that crashes on the reef. And over time, the more this event happens, the more you can have coral fragmentation happening. So essentially, those more vulnerable coral species can essentially be decimated over time. An example is mostly the branching corals, and then under increased storm frequency, those branching corals pretty much break and fragment. You have what's known as rubble fields forming. And then what you'll have left essentially is those more durable corals like the massives, such as Porites, Favites, and Favia species, which are a bit more globular in shape and do not have enough surface area contact with the water column. Those will be the ones remaining on the reefs after these high amount of damaging events have occurred. So essentially, if you increase the frequency of storm events, you're more likely to remove those fragile branching corals on a much more frequent basis from the reef. So now we've understand these two factors in general, I'm going to take you back to Heron Island, and we're going to talk about a study that I performed in 2014 that actually tests both of these effects on corals on the reef flats of Heron Island. So how did we mimic this? So we actually ran a short 12-day experiment on Heron Island during that 6-week field campaign. And our aim was to monitor coral stress and growth responses under temperature, high temperatures in particular, and injury to mimic this climate change impact. So what did we exactly do? So myself and 3 of my colleagues, we walked out onto the shallow reef flats of Heron Island, and we chose certain coral colonies to sample our corals from. And you can see Jorg in this image has a bucket with him, and we have to be able to transport the corals quickly from the reef, off the reef, to holding tanks quite quickly to prevent them from stressing out. So we sampled a branching coral, one of those fragile coral species that I was just talking about. The species is called Acropora aspera, and we sampled about 20 colonies from the reef flat. And these are going to be used in our experimental study. Now, once we sampled them, we actually transferred them into 2 holding tanks, and these tanks were able to hold about 1,000 litres of water. And essentially, these tanks recycle the water from the reef flat in about 1 to 2 minutes. So if you think of trying to replicate a normal reef condition, we need the water to be able to recycle quite quickly to these tanks. That's actually mimicking what's happening on the reef. Now, these 2 tanks were just left on the research deck with the corals in them for about 7 days. Now we do that because the corals have to sit in the normal water temperature in order to reduce their stress levels. So after them being broken off coral colonies on the reef, they need to reduce their stress levels. And we let them sit in the tanks for 7 days to be able to do this. Now, once those 7 days were over, we then started the temperature treatment. So one of the tanks was held at normal reef temperature, which is around about 24 to 26 degrees. And the second tank, we actually ramped the temperature up to just over 30 to 32 degrees, which is a high enough temperature to induce a stress response, but not bleach the coral, because Acropora aspera actually bleaches at around about 34 degrees. So we didn't want to bleach them and kill them. We just wanted to stress them. So we ended up keeping 1 tank at normal conditions and 1 tank at climate change predicted temperatures. We also then had to perform an injury event. So we had to mimic a storm coming through. So what we did, in half the corals in each tank, we actually snapped the branches off. So half of them on one side had their branches snapped off. So the top 1 to 2 centimeters of the branch was snapped off. And the other half, we actually just kept them intact and used those as the control group of corals. And now we could start our experiment. So we actually monitored the corals over 12 days. And what I actually found was actually quite remarkable and quite shocking at the same time. So how stressed were the corals under these 2 different temperature and injury regimes? So let's take normal reef temperatures, for example. So we know that on Heron Island, during March of when we performed the sampling, the normal reef temperature was about 24 degrees. So we sampled the corals over 12 days and sampled them every 2-day increments. Now, if you can see on the first image at day 2, that was the day that we actually performed the injury. So we actually snapped the branch off the top of the coral, and we don't expect any growth. But what we noticed that over time, by day 8, we start to see this first sign of growth back in the coral. And that's the apical tip, that top 1 to 2 centimeters growing back. And that continues for the next day 10 and day 12. And by the end of the experiment, the coral has fully regrown its branches again. That's awesome to see. And we also see this when we look at the coral calcification rate. So if you look at the bottom panel, that's the skeleton growing back. So essentially, the coral skeleton is growing in unison with the tissue and is actually building its skeleton to a healthy level under normal reef conditions. And that's what we want to see. The corals here are healthy and they're not subjected to any other temperature stress. We only have injury that's playing a major stress in this part of the experiment. Now, the middle panel is actually what we call protein expression. And this green protein that you're seeing here is actually called a green fluorescent protein. And that performs a very important function in branching corals in particular, because the protein actually helps to protect the tissue and the skeleton against UV damage from sunlight. So if you think, these corals can be quite exposed on the reef flats to sunlight. And what that protein is doing is actually shielding the tissue and its skeleton, protecting it, and allowing the tissue to grow back and perform a healthy amount of cell differentiation, which then helps the tissue to grow and extend, to recover and regenerate. So that's a great sign to see, is that the coral is actually expressing it to a nice healthy level. Starts to activate it around day 6, and you see it actually continues up until the end of the experiment. This is awesome. So corals under normal temperatures that we have on the reef today will be able to recover from an injury event following a storm. But what happens if we test this under climate change reef temperatures? Now, this is quite strikingly different. So when we injured the corals at day 2, we find the same effect as in the control tank at normal temperatures, that we don't expect much regeneration or growth occurring. But what we notice is that actually by the end of day 12, there is no significant growth of that apical tip that is noticeable in these images. So the actual growth response has been shunted by temperature. And we also see that in the skeleton as well up on the bottom panel. So the coral starts to perform calcification at around about day 10, but the temperatures are too stressful for the coral to continue to perform it. And therefore, as a consequence, it actually shuts down the coral's response to produce new tissue. It cannot perform an adequate amount of cell differentiation and therefore can't build more tissue to grow back that tip. In the same response, the protein expression of that green fluorescent protein actually shuts down. So temperature actually shuts down how much proteins can be expressed in the coral's tissue. And therefore the coral tissue and skeleton, which is trying to grow back, is therefore more susceptible to UV radiation, and potentially it has an amount of UV damage occurring here because it's not getting its protection from that protein. So that's a pretty dire response. So essentially the corals in the hot tank do not grow back at 32-degree water temperatures. So what is this impact, and what is it likely to have on those shallow water corals of Heron Island as a case study? So from our results, are we likely to see reef recovery? Well, if water temperatures remain at around about 24 to 26 degrees, we are likely to see corals perform a healthy amount of recovery after an injury event or a pulse temperature event. So essentially, if they do get damaged over time by an increased, by a high impact storm event, they are more likely to transition back to these complex states that we see on the left-hand side. They're able to perform a healthy amount of recovery and regeneration and keep performing natural reef growth processes. However, if we see that temperatures with climate change accelerate and become quite detrimental in the coming decades and reach temperatures of up to 32 degrees, we are more likely to see these complex reef systems actually perform what's known as a phase shift and transition to degraded reef systems, where we are seeing a high frequency of storm events coupled with high frequency temperature events, which do not allow reefs to recover back to their normal state. So we might see that the amount of corals and the species of corals that we find on this healthy reef system might be severely impacted if we transition to a degraded reef system. You might just find a couple of species which are more resilient, dominating in these degraded reefs, like parietes and those massives that we talked about, and we might not find enough growth back of those branching corals. And as a result, you're going to have a detrimental impact on the biodiversity of those reefs. So if you think about that reef structure causing or providing important habitat for vertebrate species like fish and other inverts like sea urchins, sea stars, and mollusks, you're going to have a massive reduction in biodiversity if you lose that structural complexity of the reef. So it's going to have a bottom-up effect, as we call it, on the reef structure. So are we likely to see reefs transition back? It is possible. But there is a way out of this, and it comes down to the actions of us as humans and what we can do to make a long-lasting impact on reefs worldwide. So what can scientists and you do to help? So currently, there's a number of adaptive management strategies which are being tested and troubleshooted, in particular here in Australia, to help reefs combat rising temperatures, for example. So reef recovery and adaptation can be assisted through selective evolution and gene flow. And this is a very sophisticated method by which the Australian Institute of Marine Science in Townsville are actually implementing and testing. And that's by selecting species of coral and their algae with the most resilient genetics to combat rising temperatures. Now, certain genetic strains of the algae or zooxanthellae are actually more adapted to higher temperatures, and scientists are actually selectively cultivating these algae to help artificially inseminate the coral animal to become a bit more resilient to higher temperatures. And that's producing a variety of corals that we call supercorals that are going to be able to be kind of the forefront of adaptation to high temperatures. Another option is coral transplantation on degraded reef systems. Now, this will be site dependent and depend on the regions of which transplantation is going to be targeted. So if, say, if there's sections of the Great Barrier Reef which are dominated by branching corals, you want to be able to selectively breed and cultivate corals and algae of those regions and be able to then transplant them to the same areas of which they're adapted to. So you want to be able to transplant like for like corals, okay, that are going to be a bit more resilient. And of course, the most important one that we can all do is helping to reduce carbon emissions. Now, we know that the trajectory of this climate crisis is quite exponential currently. So by reducing our fossil fuel consumption and transitioning to more sustainable and renewable energy sources, we can therefore provide better hope for coral reefs to help alleviate any future stress as a result. So on an individual basis, reducing our carbon footprint is essential. And there's small things you can do to help reduce your footprint. This can be by increasing the amount of time that you you take it home to switch off appliances and unplug them, because every bit of saved electricity helps to reduce your footprint. You could also try and take public transport more often. That can actually help to alleviate how many cars are on the road at one time. So there's small things you can do. It might not seem so significant in the broader scheme of things, but if everyone did a small part, we can make a big difference. And of course, for some people, even eating less meat is better for the environment as well, and reducing fossil fuel emissions. We know that certain greenhouse effects from methane, for example, rely on the production of meat, so such as cattle. So there's small ways where we can all make a difference to help combat the climate crisis, and therefore provide greater hope for our coral reefs to survive. But it all comes down to what we can do as individuals and as a society. So I hope you were able to kind of grasp how my career has been able to implement, certain research that can better forecast how our reefs might change as a result of climate change. And I'd lastly like to thank the Western Australian Marine Science Institution for giving the opportunity to share this. It's very important that we share our research, and particularly because this was something that I did while I was closer to your age. So it meant quite a lot to me to be able to share with you how you can make a difference if you choose to go into any form of environmental sciences in the future. So I'm going to end there